We've looked at cell division with mitosis so that multicellular organisms can grow from a zygote to a trillion-celled organism, or how a lizard can regrow its tail, or a sea star can regrow its arm, or how we can regrow or replace our skins that have sloughed off. Now let's take a look at meiosis. So we were looking at the life cycle of a human, and this process right here, we want to take a closer look at that, and that's cell division with meiosis. So we've looked at cell division with mitosis, and we got to get two identical daughter cells with a full complement of chromosomes. Now we want to look at how we get the gamete cells, so that these gamete cells can then be combined in fertilization to get our zygote. So how do we get half of our 46 chromosomes in one cell? So how do we get just 23 chromosomes in one cell? So let's take a closer look at that. How does that happen? Well, it's meiosis, and meiosis is not a cycle, because at the end we get 4 cells. So let's take a look. A lot of this is similar to mitosis, what we've already seen. A lot of the stages are very similar to what we've seen in mitosis. So interphase G1, this organism has 4 chromosomes. So it's 2N, which means it's diploid. And in S phase, again, the DNA replicates. So remember, these are homologous pairs of chromosomes, and these are also homologous pairs because they're very similar. They're the same length. They have the same genes on each of them. So our DNA is replicated, and the 2 identical strands are connected at the centromere, by the centromere. So this is also 1 chromosome right here. Okay, so that's interphase, where DNA is replicated. That's the same as it was in mitosis. Now let's get into meiosis. And we call them different. It's prophase, but it's prophase 1, and metaphase 1, and anaphase 1, and then this, oops, that's been whited out. So this is telophase 1. So this is considered meiosis 1. And then over here, we've got prophase 2, metaphase 2, anaphase 2, telophase 2. So this is meiosis 2. So let's take a look at meiosis 1. What's different? So in prophase 1, we have our nuclear membrane again starting to break down. We've got our centrosomes going to opposite poles and our spindle fibers coming out. But now, instead, these chromosomes, the homologous chromosomes, actually pair up. They hook up. They physically hook up at the centromeres. And during this phase, something called crossing over happens. Because these chromosomes are so similar that the strands on the inside, they can actually physically cross over each other. And when they do that, a piece breaks off of one and gets hooked on to the other, and a piece on the other gets hooked on to the other. And this leads to unique daughter cells. So these cells at the end are unique because of, one of the reasons is because of crossing over. Okay, so that's good for prophase. So our homologous pairs have hooked up and they've crossed over. So now we go into metaphase 1. And instead of our sister chromatids hooking up on the metaphase plate right here, it's our homologous pairs are lining up on the metaphase plate. So our spindle fibers come down and they attach to the kinetochores of each homologous pair instead of to the sister chromatids. So that in anaphase, the spindle fibers are pulling the homologous pairs apart. So these chromosomes are now pulled apart. So that now in anaphase 1 and then in telophase 2, there are now 2 chromosomes in each daughter cell. So we started out with 4 chromosomes. Now we have 2 chromosomes in each. And that's considered 1N, or called haploid. And I think of hap as half. The chromosomes have been halved. There's only one problem, is there's still the same amount of DNA. So the amount of DNA still needs to be halved. If you look, there's 1 strand there, 1 strand there, 1 strand there, 1 strand there. So 2 long and 2 short. That's what we had in the beginning, 2 long and 2 short. So the amount of DNA now must be halved. And the rest, the meiosis 2, now looks a lot like mitosis. Okay, so here we are in telophase 1. Cytokinesis happens again, and the rest of the cell is now divided in 2. And then we go into prophase 2. So we've got our cells, same thing. Actually, this looks a lot like mitosis. So our nuclear membrane starts to break down again, and our DNA starts to condense again. It starts to package up. In between, it did actually go back to chromatin form. So it packaged up and gets condensed again. And our centrosomes move to opposite poles. And then in metaphase 2, this is just like mitosis, our sister chromatids line up on the metaphase plate, and our spindle fibers come down and attach to the kinetochores on the centromeres of our sister chromatids. And then in anaphase, the spindle fibers pull the sister chromatids apart, and now they're called, just like we said before, they're now called daughter chromosomes. And now we go to telophase 2, where our nuclear membranes start to form again. And now we have 4 daughter cells, each with now 2 chromosomes and we have half the amount of DNA. So these are now gametes. And in a male, these would be sperm, and they would hook up with an egg to form a zygote. Let's see, so a couple of things we need to make sure that we understand. So there are, in meiosis, there's two divisions. There's the division that's happening here in meiosis 1, and then there's also another division over here that happens in meiosis 2 to get 4 cells. So the cell has to divide twice. So 2 divisions. What else? Okay, the source of diversity. So we talked a little bit about that when we talked about crossing over. So the sources of diversity, we mentioned that these gametes are unique, not only because they have half the amount of chromosomes, but because of crossing over. You can see that the DNA, these chromosomes aren't exactly like the chromosomes that were in the parent cell. So we have a source of diversity in the population because of, 1, crossing over, and 2, the other place that we get diversity is the way that these homologous pairs line up. They can line up in many different ways. And if you think we have 23 homologous pairs, 46 chromosomes, they can line up in many, many, many different ways. So that's also another source of diversity. And that's called independent assortment. Let me make sure, whoops, try that again. We want an N right there. So independent assortment. Okay, so 2 sources of diversity here. Here's 1, and independent assortment is 2. The other way of getting independent assortment is if we look back at our life cycle. Fertilization will also give us diversity. So there could be many, many different combinations between egg and sperm to come up with the full complement of chromosomes in this individual. So many, many different combinations there. Okay, so that meiosis is important for having unique daughter cells to be able to provide a source of diversity for evolution and for natural selection, for the mechanism of evolution, the natural selection to work. It needs this diversity in the population. Okay, so I hope you found that interesting, and we'll see you next time.